that. Like a Halloween prank or something. So photosynthesis is what we're talking about today. If you were with us last week, we talked about cellular respiration. The good news is cellular respiration and photosynthesis share a lot of commonalities. And so students oftentimes find that, I'm sorry, last week we didn't talk about cellular respiration, did we? I'm blanking now. Can't remember if we talked about cellular respiration or cell signaling, but regardless, with photosynthesis and cellular respiration share a whole heck of a lot of similarities. And so students always, or seem to find that learning photosynthesis after learning cellular respiration makes it much more straightforward. And there's a lot of inverse properties that happen. So hopefully this isn't as challenging as cellular respiration maybe was, and I'll be able to explain some pretty cool things. All right, so let's just start out with the general purpose. Why do we care about photosynthesis? And most people are actually more familiar with this process than cellular respiration, but basically we are converting light energy. We are converting photons of light into usable chemical energy. In simple terms, we're taking light and we're turning it into the most common example, glucose. Okay, and then that glucose will go on to do lots of wonderful things for the plant and for us as well. So that's the end goal there. So you'll see our reactants on the left-hand side. We have carbon dioxide and water, right? And we'll talk about where those come from, what they're used for, etc. And then we also have this sunlight coming in, which is obviously a very important part of this reaction. And that's going to give us glucose, which is going to be our energy that we come out with, as well as oxygen, which we're going to find out is really just a toxic byproduct for the plants. Obviously isn't for us, and it's one of the main reasons why we care about photosynthesis, besides the fact that it also produces all energy that we have on Earth. So lots of big things here, very important process, and again, by the end of this, we'll be able to talk about where everything came from, where everything ends up, and the major purpose of that thing. And if you can understand those big concepts, you will be good to go with photosynthesis. All right, so here's what's going down. We have two major steps here. So in cellular respiration, we had three. We had glycolysis, the Krebs cycle, and the electron transport chain. In photosynthesis, we only have two. We have the light reactions, which are actually going to involve an electron transport chain and lots of different proteins and things like that. And those are dependent on light. That's why we call them the light reactions. Light-dependent reactions is another way to say it. And then the second one is the Calvin cycle. Some people may also call the Calvin cycle the light-independent reactions because they're not dependent on light. They're dependent on the light reactions, but not on light itself. It can happen at any time is basically what's going on there. And we'll talk about the differences there. I'll show you some diagrams and we can do some practice with that as well. All right, so the light reactions. Whew. We're going to stay on this picture for a while, so get comfortable here. But super important things happening here, and I want to make sure that we're able to walk through every part. Now, I also want to clarify, you are not going to have to label a diagram like this or do something like that on the AP exam, but you will need to know where things come from and where they're going. And truly the best way to know where things come from and where they're going and what they're used for is to just know what the process is, right? Otherwise, you end up just shortchanging yourself. So we're just going to go all in. I will summarize the most important parts for you, but you'll be able to see kind of this overall process. Okay, so the first thing that I will mention is that we have a lot of really, really important components here. The first two are these big green blobs. We have two of them labeled PS2 and PS1, and that stands for Photosystem 2 and Photosystem 1. So I'll write that out for you. Photosystem 2 and PS1 would be Photosystem 1. And a question that you might be asking yourself, and rightfully so, is why in the world then is Photosystem 2 before Photosystem 1? That's a very valid question. Basically, scientists discovered this guy, Photosystem 1, first. So instead of renaming them when they realized that it actually came second to another photosystem, they just decided to make all of your lives miserable forever. No, but that's really the reason. So photosystem 1 was discovered first, photosystem 2 came next, and they just kept it that way. So these photosystems, they are covered in chlorophyll molecules, and chlorophyll is a pigment. Okay, you may have heard of it before. My favorite reference is the Billy Madison quote, "Chlorophyll, more like borophyll." If you're not familiar, Billy Madison is a phenomenal movie. But what chlorophyll is it is a pigment, and it is what actually gives plants their green color. So cool thing about color and the way that we perceive it, we perceive the reflection of light, right? So these really high energy pigments that are sitting on these photosystems, they absorb predominantly red light. And you can see this, it's actually at wavelength 680 nanometers and then wavelength 700 nanometers. So that's the light that they're absorbing. It's in the red spectrum, but then they're reflecting green light or kind of everything else, which is going to appear green to our eyes. So it's absorbing red, it's reflecting green. That's what's hitting our retinas. And that's why we see bright green leaves. Except for now in fall, because the leaves are dying. It's well, we're going to talk about it, but it's a advantage for trees to drop their leaves in the winter when there's less precipitation, because the leaves tend to allow for a lot of water evaporation, and so they kind of just hunker down and hibernate in the winter. Hence the leaves dying, and when they're dying, their pigments are not there anymore, and we have all the pretty colors of fall. All right, I'm giving you a lot of extra knowledge today too. So we're going to start. So as I said, photosystem 2, photosystem 1, both very, very important proteins that are sitting in the membrane. And I should have contextualized this a little bit, but we're in the chloroplast, right? So the chloroplast is the organelle in which photosynthesis occurs. Inside of that chloroplast, there are things that look like little pancakes, right? There's really no other way to describe them. We call those thylakoids, and that word's down here for you. And that is where all these membranes are located in these little pancake forms. And so that's where we're actually seeing this part happen. And then the stroma, which you can see up here, is kind of like the syrup, if you will, or the gooey stuff outside that fills up the rest of the chloroplast. And that's where the Calvin cycle will take place. So there's all your context. So what's going on here is light is coming in, a photon of light from 60 million miles away from the sun. Light, if you know anything about it, is both a particle and a wave at the same time, which is nuts. But it comes in, and a photon of light will strike the photosystems. So we're going to start by talking about photosystem 1, because I can't talk about them simultaneously. I'm sorry, photosystem 2, the one that comes first, because I can't talk about them simultaneously. And when this photon of light comes and hits photosystem 2, it hits a chlorophyll molecule, and an electron on that chlorophyll molecule gets really excited, right? Think about light as an energy source, right? And so when photon strikes the chlorophyll, it's transferring its energy, and that electron gets really excited. It actually moves up in its energy state, and it will essentially pop off a photosystem 2. So light hits, electron gets excited, electron pops off photosystem 2, okay? That electron is going to go do wonderful things for us, but in thinking about what is left behind, we have a photosystem 2 molecule that is without an electron, which is a big problem, right? Because if you've taken chemistry and you know about radicals, right? When things lose an electron and don't have a full complete set anymore, we can have a lot of chaos ensue. So what happens here is we need to replace, I always think of photosystem 2 as like a parent whose child has left for college, right? The electron is that child, it is leaving us, and the parent needs to fill the void left by that lost electron, okay? Or more technically, we have like a free radical and we need to replace it. But so what we're going to do then is we're actually going to split a water molecule. So this is where water comes in. Remember that water is one of the reactants of photosynthesis. So we're going to actually go ahead and split a water molecule that's chilling in the thylakoid. And when we do that, we will release an electron, right? We're going to release some oxygen, we're going to release some hydrogen, and we're going to release some electrons. We're going to use one of those electrons to replace the one that was lost. Okay, so I'm going to summarize that again. I'll do this pretty frequently, like pause us and renorm. So photon of light hits a chlorophyll molecule on photosystem 2. Electron from chlorophyll molecule gets super excited and leaves. Photosystem 2 is now without an electron, so we replace it with one from water. When we split that water to take its electron, we form some oxygen, some hydrogen, and that's great because oxygen is a really great byproduct that we would not survive without. Okay? Any questions about that part? Feel free to write anything in if you're like, what in the world? This is also, I think I said the same thing when I was going through cellular respiration. This is one that like deserves a rewatch, right? Just so that you can rehear what I'm saying and make sense of it all. Okay, but again, photon hits photosystem 2, electron hops off because it gets really excited. We replace that electron with an electron from a water molecule. When we split that water molecule to take its electron, we form oxygen and hydrogen. Really convenient byproduct. Okay, all right. Similarly to carbon dioxide, for us, oxygen is actually going to be a toxic byproduct. So too much oxygen can actually kill a plant. And so this oxygen is just going to leave the plant. It will leave the plant through its stomata, little holes in its leaves. It'll get released into the atmosphere. We will breathe it in. We live to see another day. Okay. These hydrogen molecules are going to do something for us a little bit later on. Think cellular respiration concentration gradient, but we'll get to that in a second. All right. So we've replaced photosystem 2. It is whole again, but this lost electron, as I said, is destined for great things. And basically what it's going to do is it's going to travel through an electron transport chain, very, very similar to the electron transport chain we learned about in cellular respiration. So I'm not going to go into too much detail with this because we've already talked about it, and you can go back and rewatch that video if you need a little refresher. But that electron travels through this electron transport chain that's going to end up on, eventually, photosystem 1. Okay, so all of these things in the middle, you don't need to know their names, but they're all different proteins that are in the electron transport chain. If you remember, they kind of get pulled through. Electrons kind of get pulled through like they're on a conveyor belt, and this very high energy electron that has been hit by a beam of light is going to provide us with some energy to pump some hydrogens across the membrane. Okay, same exact concept as it was in cellular respiration. No difference here, right? We have an electron, it's traveling through the electron transport chain. We are pumping hydrogens against their concentration gradient from an area of very low concentration of hydrogens to an area of higher concentration of hydrogens. They don't want to do that, but we use the energy. Sure, guys, I don't know what it is about talking after 6 p.m., but it's making me yawn. And so we're going to use the energy from this little guy. We're going to pump those guys across, and he will end up on photosystem 1. Okay, starts at photosystem 2, pops off, goes to the electron transport chain, ends on photosystem 1. All right, let's talk about all these hydrogens then, and then I can take time for questions if there are any. So all of these hydrogens that have been pumped into the inner membrane and the ones that came off of water, there's a really high concentration of them in here, and there's a low concentration of them out here. And guess what? It's our old friend ATP synthase. Okay, remember anything that ends in ACE is an enzyme. So this is the enzyme that synthesizes ATP, right? Same exact process as in cellular respiration. Hydrogens get pumped through ATP synthase, and the kinetic energy from that process allows us to phosphorylate or add a phosphate to ADP, which gives us ATP, okay? I'm going to write those vocab words up again in case you weren't here for the cellular respiration. Phosphorylate is to add a phosphate, and ADP is adenosine diphosphate, so 2-phosphate. ADP is adenosine triphosphate, 3-phosphate. Cool. Okay, so we do that. That's wonderful. We're going to use all of those ATP in a little bit in the Calvin cycle. So really the point of the light reactions is to make these high energy products to be used in the Calvin cycle. Okay, but again, this ATP gets made through ATP synthase because of this concentration gradient formed by the electron transport chain. Okay, all right, last step and last product here We also have, right, we have light hitting photosystem 1. Same exact thing happens as with photosystem 2. Hits a chlorophyll molecule, electron gets really excited, electron hops into a higher energy state, kind of pops off of photosystem 1. Thankfully, we don't need to replace this guy because he's already been replaced by the electron lost from over here, right? The electron that went through the electron transport chain is there to replace, okay? But this electron that was lost then is going to go up here and reduce NADP+. Okay, so key player here, if you remember NAD+ from cellular respiration, which was an empty electron carrier, okay, NADP+ is just an empty electron carrier in photosynthesis. You can think P for plants or P for photosynthesis, okay? So when NADP meets this electron that's popped off of photosystem 1 and some of the hydrogen ions that are floating around, we form a full electron carrier, which we call NADPH in this instance. Okay, so any questions? I can run through that one more time, but if there's specific questions that people have, I would love to answer. No? Everyone's too busy eating their candy. We don't want to ask you questions. All right, I'll run through that one more time. Light hits chlorophyll molecule in photosystem 2. Electron gets really excited, runs through the electron transport chain, ends on photosystem 1. In the process, it pumps some hydrogens across the membrane, which are used to make ATP. Lastly, well, not lastly, but to make photosystem 2 whole again, we split a water molecule, we rob it of an electron, and that guy is good to go. This guy, who has also lost an electron, uses this electron to reduce NADP into NADPH, and that electron had already been replaced from photosystem 2. Okay, big takeaways here. Water is used to replace an electron. We call it the initial electron donor. It's kind of ironic because if you remember kind of the reverse of this reaction, oxygen is the final electron acceptor in cellular respiration, and it grabs onto that electron and some hydrogens and makes water. And so this time it's the initial electron donor. It's giving away its electrons and we're making oxygen in return. So that's one big takeaway. Other big takeaways would be the production of these really high energy ATP and NADPH molecules, which are going to come in handy. All right, so there it is for you. Light, water, ADP, and NADP+, because remember we need these like empty shells, these like empty batteries and empty electron carriers coming in. And we'll talk about where they come from in a little bit. And what comes out, ATP, which is going to be really, really useful, electron carriers, those NADPH molecules, and oxygen, which the plant sees as a toxic byproduct, and we see as the best thing ever because now we can breathe and live another day. All right, there it is, the light reactions. Okay, so all of these things, so I should say where these things are destined for. So ATP and the electron carriers are going now to the Calvin cycle, okay? Because the end goal here is that we're converting light energy into not ATP and electron carriers, but into glucose, right? Which is going to be a higher energy molecule that we can then store as well. So that's super important. So that's where they're ending up, and we'll talk about why in a second. But then the oxygen just gets released. Okay, Calvin cycle. All right, a couple important things to know here. One, do not panic. You do not need to know most of these things, which is great, okay? But you do need to know a few things. First of all, you need to know that the first step here, so in the Calvin cycle, basically what's happening is we're taking CO2 out of the air, right? And then we are going to add it to and combine and split apart and do all sorts of things with other carbon structures that already exist in the plant. And eventually we are going to have something called G3P, which is going to be the precursor to, which you see in the bottom left-hand corner down here. And this, or sorry, down here. Why does that darn thing show up? It needs to go away. Anyway, G3P, which you see down here, which is going to go ahead and make glucose. And it can also make some other products too, and I'll talk about that in a second. Okay, but here's our second part here. So CO2 is going to be taken out of the air, right? We obviously exhale CO2, so do all other animals. And and everything that is going through cellular respiration. And so that gets taken out of the air, and it gets taken out of the air and gone through this process called carbon fixation. Okay, so that's kind of an important one to know. Basically, this is the incorporation of this gas, CO2, into an organic molecule. So that's kind of crazy to think about, that plants are utilizing thin air to make sugar. It's crazy. But they're going to do so with the help of this enzyme Rubisco, okay, which is kind of a good one to know because sometimes it pops up. Ribulose bisphosphate carboxylase is the full name, if you're wondering why it doesn't end in ase if it's an enzyme. It does. This is just its nickname. But Rubisco is going to be the enzyme that's responsible for fixing this carbon. Again, carbon's not broken, but we're just taking it from a gaseous state of carbon dioxide, and then we are incorporating it into an organic compound. Okay, so that's an important thing to know. Carbon fixation, Rubisco, etc. Another important thing to know is to see the fact that we're using tons of ATP and NADPH to go through this process, right? And you can see that as you go around the circle. And if you think about it, right, if we are taking carbon dioxide out of the air and we're turning it into a really high energy molecule like glucose, you better believe it's going to cost energy, right? So it's going to cost ATP. And we're also making and breaking a lot of bonds. So it's going to cost some electrons as well, right? Remember, electron carriers will drop off electrons. Electrons are used to help make bonds. And so we're going to need both of those things, which is why the light reactions are so important. We're going to need both of those things in order to make this high energy G3P molecule, which will go on to make glucose. Okay, so key points here. Carbon dioxide is going through something called carbon fixation, where it comes from a gas and gets turned into an organic compound with the help of an enzyme called Rubisco. And then this whole process, which you don't need to know all the steps of, thank goodness, involves lots of energy and lots of electrons in order to make a really high energy product called G3P, okay, glyceraldehyde 3-phosphate. But the end goal there is going to be that G3P then will go to make glucose, which we know and love. So G3P can also go to make cellulose and starch and lots of other things for structural or storage purposes. So it's a really great product here, but we just kind of usually think of it as making glucose because that's what we talk about most when we talk about photosynthesis. So Calvin cycle is a lot simpler than the light reactions. Well, it's not, but the parts that you need to know are kind of simpler. And the way that it's set up is a little simpler. So what goes in? Carbon dioxide from the air, okay, ATP and NADPH from the light reactions. So that's where all that comes from. What comes out? Glucose, G3P or glucose, right? And then also, and I should have showed you this on the slide, we're recycling all of these guys. So if you remember, we talked about recycling of electron carriers and ADP a lot when we talked about cellular respiration and how important that is because we have a limited number, right? And what we're doing here is we are recycling those things. So then these guys can go back to the light reaction to be refilled, okay? So that's actually a really important product of the Calvin cycle, even though like an empty battery and an empty electron carrier seems kind of useless. It's not, because if we didn't have this recycling process, our light reactions would eventually stop because we would have nothing else to kind of fill up. Okay. Here's your kind of like general overview then of everything that I just said. So we have these energy building reactions or the light reactions where we have water going in, we have sunlight going in, the water will get broken apart to form oxygen, oxygen gets released, okay? And then we form these really high energy NADPH and ATP molecules. They are headed for the sugar building reactions, also known as the Calvin cycle. In the Calvin cycle, we're going to use these things and we're going to convert CO2 in a very complex process into sugars like glucose, okay? And we're also going to recycle those ADP and NADP back into the light reactions. So this diagram I find very helpful. So it's like a really good cheat sheet. And if you kind of know where the parts are coming from and where they're going, then you'll be in really good shape. Really important distinction to realize that CO2 and O2 are not related in this process either. I know I mentioned that many times with the cellular respiration, that CO2 is the byproduct from the breakdown of glucose and has nothing to do with oxygen. Same thing here. Water breaks down and forms oxygen. CO2 and oxygen are not involved in this process. They're actually in completely different sides of the reaction. So yeah, they're not involved. Okay, so we're going to look at a few practice problems then. So that's really it as far as like big picture things you need to know about photosynthesis, which is good news. But I want to make sure that you get some usable practice because that's really, really helpful. So this is a kind of a lab setup that has some multiple choice questions that go along with it. It's a lab that maybe you guys will do. I actually did it with my class this year, but it does show up oftentimes on the AP exam. So I'll give you a second to read through this. Okay, hopefully that was enough time to read through. And you may be thinking to yourself, what in the world? There's a lot going on here. And there is. But we can break it down, and it's good practice with like a high level AP question where you may have to break it down. My usual strategy for this, if this was on paper, which is what I like to do with my class, is just to start drawing out things for myself. Okay, so the first thing that we need to figure out is this thing DPIP. So DPIP is a chemical that is blue and becomes colorless, right? This is what it says in the passage, when reduced, and it's used as an electron acceptor, okay? Same thing as an electron carrier. Now, we need to know what reduced means. So I know we've talked about this, but oxidation, right? Oil rig, oxidation is loss and reduction is gain. And I can write that into the chat box again, but I know we've talked about this. Oops. Okay, and so if something is reduced, then it means it's gaining an electron. Okay, basically when DPIP, or when this electron carrier fills up, which it does in the light reactions, it turns clear. Okay, so what this tells me then is if photosynthesis is occurring, then this blue dye will turn clear because electrons will have been picked up by it, okay, from photosystem 1. So that's thing number one. The second thing I have to think about is transmittance. Transmittance is the percentage of light passing through something. So then you have to think to yourself, okay, is more light going to pass through a blue substance or a clear substance? And the answer should be clear substance, right? So things that do photosynthesis are clear. So things that are doing photosynthesis more are going to be clear, are going to have a higher level of transmittance because more light can travel through that substance. Now that we know that, these next following questions shouldn't be too hard. Okay, so number one, after 30 minutes, which cuvette would be expected to have the highest level of transmittance? All right, so on first glance, it looks like cuvette 1 and cuvette 2 are identical. But if you were reading carefully, you will read that cuvette 1 was kept in the dark. Okay, so this guy we can eliminate right off the bat because we should not see the light reactions happening here if there is no light. Okay, so we're eliminating cuvette 1. We can also go ahead and eliminate cuvette 4 because there's no chloroplast in that solution. Sometimes my students get tripped up because it says 3 drops of water, and so they see more water and they think, ooh, more photosynthesis. But there's no chloroplast, right? So if there's no chloroplast, we're not going to see photosynthesis occurring. Okay, so we're down to cuvette 2 and cuvette 3. Hopefully you've already come to this conclusion yourself, but it should be cuvette 2 that's going to have the highest level of transmittance because that one has unboiled chloroplasts as opposed to cuvette 3, which has boiled chloroplasts. Now remember, if we are boiling something, we are denaturing, right, or breaking down all of the proteins in that substance, okay? Denaturing meaning like unraveling or unwrapping the quaternary or tertiary structure of that protein. Those things are not going to function anymore. Okay, so that's what boiling does. So that guy, we shouldn't see much photosynthesis at all. Maybe a little bit, like I did this lab with my students, and cuvette 3 did have some color change, but mainly because I don't think I boiled it long enough. So, or sometimes just not all the proteins denature. Okay, awesome. So your best answer choice here is B, but we're going to use this setup for the next 3 questions, so stay tuned. All right, question 2 is very easy, or should be at least if you've been paying attention. Which of the following molecules is DPIP replacing in this experiment? Well, remember we just talked about it and it was listed in the passage. DPIP is an electron acceptor. So the thing here that accepts electrons for us is C, NADP+. Okay, so those are some easy questions that you could get lots of points on the AP exam if you're familiar with this process as a whole. Okay. Question 3, which of the following best predicts the expected results for cuvette 1? So remember, cuvette 1 is kept in the dark. So take a minute to read through the answer choice options listed, and then we'll talk about which one is the best answer choice. All right. Hopefully you were able to use some process of elimination and come up with D as your best answer choice. So what we need to think about here is that this dye, DPIP, is blue unless it is reduced, unless it's gaining electrons, which would happen if we were in the midst of a light reaction. Okay, because this guy is not in the light, we shouldn't see anything happen. Okay, gradually increasing transmittance would mean that photosynthesis was occurring, right, which it won't be. And then gradually decreasing or increasing then decreasing. Decreasing would mean that we are getting more blue. But whatever we're starting at is as blue as we're going to get, right? That dye is not going to get any more condensed. So it should be D, that transmittance level should just remain the same. It's like a really good control group to compare everything else to, okay? But sometimes students like try to draw trends that don't exist. Sometimes things just aren't supposed to happen, so you have to get comfortable with that. All right, lastly, question 4 is a little tricky. So the percent transmittance for cuvette 3 as measured by the spectrophotometer, sorry, forgot a few syllables there, are shown in the table above. Which of the following most likely explains these results? So the first thing that we need to do is figure out what cuvette 3 is. We'll notice that cuvette 3 is that one that has the boiled chloroplast in it, okay? So that's going to give us a helpful hint there, okay? And you'll also notice that over time, our percent transmittance is increasing. And if you remember back to what we were talking about in the beginning, when we reduce that DPIP dye, it gets more clear and we will see that increased percent transmittance. So something's going on here because tube 3, right, we have boiled chloroplast in. We would assume that the proteins are denatured, and yet we're still seeing an increased percentage of transmittance, which means we're still seeing some photosynthesis occurring. Okay, so all of that is a conclusion that I would draw before I even looked at any answer choices to make sure that I'm staying on the right track. So I'll give you about a minute to read through the answer choices, and then we'll talk about the best answer choice. Okay, hopefully you were able to eliminate a few answer choices. So B, these results are expected based on the presence of cuvette 3 in the light. We should be able to eliminate right away because it's not accounting for the fact that the chloroplast present in the solution was boiled and therefore the proteins should be denatured. D also is an extrapolation, or it's something beyond the scope of the study. We're like making assumptions then, and the AP exam will often put something in that looks like we're making an assumption. Don't make assumptions, ever. Okay, so it says the light intensity to which cuvette 3 was exposed was inconsistent during the course of the experiment. There's nothing in that table that says that is the case, right? And so that one can be eliminated as well. It also is beyond the scope of what we're actually trying to figure out about those boiled chloroplasts. Okay, so our answer choices we should have between A and C. A says these results are expected since not all of the thylakoid membranes were disrupted by boiling, nor were all of the enzymes involved denatured. And C says the chloroplasts were inadequately heated. Boiled chloroplasts should have a higher level of transmittance due to the increase in molecular movement. So a lot of my students will immediately want to choose answer choice C because it says the chloroplasts were inadequately heated, which is probably what happened here, hence the increased transmittance. But the second half of that sentence is wrong. And so I always like to highlight this question because it shows a really easy mistake that students oftentimes make, which is grabbing on or latching on to something that seems right at the beginning. But the second half of that sentence is not correct. It's not what we would expect from a boiled chloroplast. Okay, so your best answer choice here is actually A. Were these results expected to us beforehand? No, but the explanation for why they would be expected is correct. Okay, not all the thylakoid membranes were disrupted by boiling, nor were all the enzymes involved denatured. Therefore, these results are expected. That is the only answer that successfully makes sense with the science that we know, as well as the information in the table. Tricky question. All right, we'll go through, these are actually our last two. So we'll go through these last two, we'll wrap up a little early. Maybe you guys can get some trick-or-treating in, who knows. So question 5, this is just a good thought question, and it's another one that kind of highlights the idea that you might need to write out or draw out kind of a picture for yourself, like we talked about with DPIP and the color change, so that you understand what's going on in an experiment. I do that for myself constantly during the AP exam, so I'd suggest that you do the same just to keep it straight in your brain. So question 5, carbon dioxide is passed into a solution of bromothymol blue indicator until the blue acid solution turns yellow. So if I was writing this down, the first thing I would write is blue to yellow, and then above the arrow I would write CO2, right? Because that's the thing, carbon dioxide is the thing that's turning the solution from blue to yellow. That way I can keep that straight in my head. A sprig of Alodia, an aquatic plant, is then placed into this yellow solution. After a few hours in the sunlight, the yellow solution containing the Alodia turns blue. Why did this happen? So I'll give you like 30 seconds to think about it on your own, and then I'll talk through the answer with you. Okay, so the best answer choice here is that the plant is using or absorbing the CO2 from the water and converting it into glucose via the Calvin cycle. So the reason why I like drawing that picture out so much is because if you have blue plus CO2 yields yellow, then you can kind of reverse that and say, okay, well, how are we going to get back from yellow to blue then? We're going to have to subtract that CO2 that we added. Great news, plants do that all the time, right? They're taking it out of solution and then they're converting it into glucose. If we're removing all that CO2 from the solution, then it will naturally turn back to blue. Someone just asked a question, Vera. So I'll start answering this really quick. Transmittance is how much light is able to make it from one end to another end. So what happens here is we'll put like a little clear container inside of a machine called a spectrophotometer, and we'll shine light through it and then monitor on the other end how many of those light particles made it through. Okay, so if you think of like the transmittance of my finger, light is not really coming through my finger at all. Maybe like a teeny tiny bit, okay? But so the transmittance here would be very low, would be 0%. The solutions that we were dealing with in the lab were just not like actually plant solutions, just chloroplast added into those solutions. And so what we're seeing there is just how much light is able to get through that mix of solutions. And if it's blue, less light will be able to make it through the other side, more light will be absorbed by that pigment, as opposed to if it's clear, when light can easily pass from one side to the other. So transmittance just means how many light molecules are we getting from one side to the other, the higher the transmittance. In this case, the more photosynthesis occurred because that dye was becoming clear instead of blue. Okay, last question before I leave you for the night. This is another good one, kind of a challenge thinking question. So the Calvin cycle requires ATP and NADPH, the products of the light reactions, right? We spent a lot of time talking about that earlier. If a classmate asserted that the light reactions don't depend on the Calvin cycle, and with continual light, we could just keep on producing ATP and NADPH, how would you respond? So we know the Calvin cycle depends on the light reactions, right? Because we're grabbing the products from that to use the energy and electrons to form glucose. But this student is saying that the light reactions don't depend on the Calvin cycle. Only the Calvin cycle depends on the light reactions. So I can tell you that the student is wrong, but I want you to think for a second as to why they are wrong. Why would the light reactions actually depend on something that the Calvin cycle produces? Think about it for a second and then we'll chat. Okay, so the main thing here, and we talked about this earlier, is the whole idea of recycling those electron carriers and those ADP molecules so that they can be refilled by the light reactions. So if we go back to this slide, you'll see that recycling process in the middle here. We're forming really high energy ATP, we're stripping it of its energy, and we're returning the uncharged battery ADP back into the light reactions to be refilled. Again, if we did not do that, we would not be able to continue to produce ATP and NADPH because we don't have the building blocks necessary to keep refilling them, right? And the same thing is true with NADPH. We are using all of those electrons to form bonds in the Calvin cycle, but then this empty carrier, NADP+, can go back to be refilled by a new electron from photosystem 1, and the cycle can continue. Otherwise, that photosystem 1 electron would have nowhere to go, that ADP would not be able to be refilled, and we would cease to have this reaction occurring. Okay, so yes, the light reactions, even though it might not seem like it, are very much dependent on that ADP and NADP coming back from the Calvin cycle once it's been used. Okay, all right, so those are the kind of like styles of questions that you will see. Looks like I have, a couple more questions. So Vera asked a good question, why does ATP turn back into ADP towards the end of the Calvin cycle? That's because we're breaking the bond. So remember ATP, adenosine triphosphate, all of its energy is living in that third bond. When we break that bond, which means we're releasing energy because we're making this high energy glucose molecule, we're left with ADP, adenosine diphosphate, and that phosphate. So all we're doing there, the reason why it's turning back to ADP is because we're breaking that bond to release that energy to use it. Another really great question about C3, C4, and CAM plants. I'm actually going to postpone this one. Oh, well, no, we can talk about it. So there's a couple of differences here, and the differences are mainly in how we are going to photosynthesize and in which capacity and the timing of that. Let me see if I can find a diagram really quick. Stay tuned, but it's way easier to show you that way. C3 are going to be our normal plants, like what I just described. CAM plants are going to be like our desert plants, and C4 are kind of in between. So let me pause really quick, and I will show you a diagram of this. Give me 1 second. Try to add it to the PowerPoint too, so then you guys have it in the future. Here's a good one. Okay, sorry, y'all. There we go. Doesn't look centered, but that's fine. Okay. Cool. So C3 plants, that's the process that I just described to you, right? So we're taking carbon dioxide, and this all has to do with the Calvin cycle, but we're taking carbon dioxide in and converting it into glucose, easy peasy. Cam plants on the other side, and these are a lot of our cacti and things like that that live in the desert, they will only go through carbon fixation at night, okay? So they go through, they take carbon dioxide out of the atmosphere and convert it into usable products only at night, okay? And the reason that they're doing that is they only want their stomata, the holes in their leaves, or if it's a cacti, like on their skin, to be open when less evaporation is occurring. So when it's really, really hot in the heat of the day in the desert, they don't want their stomata open because water is just going to leave them at rapid speeds, and they can't afford that. So they will only open their stomata at night, and that's when they'll fix the carbon in order to conserve water. And then they'll kind of keep it in this compound called C4. It's like a 4-carbon molecule, if you can figure that one out. Then they'll kind of reconvert it back into CO2 to use in the Calvin cycle during the daytime. But then they don't have to have their stomata open to the exterior environment, so they're not losing water. Very similarly, C4 plants basically do the same thing, but they just do these processes of carbon fixation and the Calvin cycle in 2 different parts of the plant. So this is also a lot of high energy plants, like slightly more evolved plants that are in hotter, drier climates. Actually, corn is a C4 plant, and basically it's similar to the cam plant. It's converting, it has like this in between, it makes this C4 in the mesophyll of the cell, and this also helps them to save some water because they can kind of convert that quicker in one condensed area, and then it moves into the Calvin cycle to be converted into glucose. And again, it's just protecting from having its stomata open too long. Just to